Well, it's my honour to introduce the next swag of speakers. And the first person in line is my close colleague, Dr. Selma Klanten. So it's my pleasure to introduce her and give you her very short bio, but very extensive. So I've known Selma for many years, and she completed her PhD in 2004 at James Cook University in Northern Australia. Her research in general explores behavioural, evolutionary and ecological patterns and processes using marine organisms and model systems. So she employs molecular tools to investigate a wide range of evolutionary processes, including phylogenetics, relationships, phylogeography, species diversification and connectivity of populations. And today she's going to present her research paper concerning the genomic and morphological evidence of subpopulations in weedy sea dragons in Australia. So Selma, I'll leave it over to you. I'm not sure if you're going to present live or if Jean is going to put up your movie, but I'll step back.
Yes, I would prefer the recorded one, Jean, if that's possible. And thank you so much for this organization, Jean. This is my first signated conference. And I'm new to signateds. As Dave said, my background is in coral reef fish. Anyways, I love these last 3-4 years that I worked on them. So yeah, if you could play my recorded one, please.
Of course. Okay.
Thank you.
Oh, welcome. I'll give me some seconds.
Thanks, Jim.
Okay, I'll show the video now.
Hi, my name is Selma Klanten, and today I will present our findings from our recent paper N+1, genomic and morphological evidence of distinct populations in the endemic weedy sea dragon Phyllopteryx taeniolatus along the east coast of Australia. Before I start, I'd like to mention my co-authors and collaborators. Myself and David Booth are from the University of Technology, Sydney. Michelle Gaither and Sam Greaves from the University of Central Florida. John Turnbull from the University of New South Wales, but he's also director of the Underwater Research Group. Katie Mills from National Parks in Victoria and Rob McKinnon from National Parks in New South Wales, Chris O'Keefe from URG. The study was funded by the SeaWorld Research and Rescue Foundation. David Booth already introduced and talked about the ecology and distribution of sea dragons. Just to recap, the weedy sea dragon, as we like to call them, are endemic to the temperate coast of Australia. So along the east coast from central New South Wales to Tasmania, South Australia, which is Victoria here, and Southern Australia, all along the Great Australian Bight to Western Australia and Perth. This region is also known as the Great Southern Reef. Currently on the IUCN Red List, sea dragons are listed as least concern, but with the caveat that populations are decreasing. They're protected in Australia under the Commonwealth Act of 1999. The preferred habitat is along a kelp or seaweed sand interface and provides shelter and camouflage, and their preferred food source are mysid shrimps. Our student Sam Allen will talk more about the habitat preferences. The males carry the eggs under the tail, as you can see here from the Sydney sample. And they do show high site fertility, so they do not move much from homes as they are slow swimmers. We know from a study by the Wilson Group based on 2 mitochondrial DNA markers that sea dragons have a strong population structure, especially the southern and the western populations. Along the east coast, haplotypes were also shared. They also recorded increased genetic diversity for the southern and western populations, but somewhat lower genetic diversity for the eastern populations. These distinct populations were also supported by quite high Phi-ST values, as you can see here in the graph. So our aim was to further resolve the genetic structure, especially for the eastern populations, using whole genome approach, not just mitochondrial DNA, and also check genetic diversity in these eastern populations. In addition, we also used external morphology measurements to compare eastern populations, mainly New South Wales populations, to the southern Victorian populations. We had 3 years of field season, and we sampled in 3 states, 5 sites in New South Wales. So 4 were from central New South Wales, 1 from southern New South Wales, 2 sites in the south, in the Mornington Peninsula, which is close to Melbourne, and 2 more sites in Tasmania at the east coast. We collected tissue from 82 individuals, including tissues from 10 washed up dead seadragons. So overall, we had 48 that included 10 dead from central New South Wales, 10 from southern New South Wales, 15 from Victoria, and 9 from Tasmania. For the morphology, we had a total of 45 samples, 20 each from New South Wales and Victoria, and 5 from Tasmania. The data was collected mainly by my collaborators, as you can see here. For the genomics part, we used double digest rat sequencing library. Preparation and sequencing was done at the Australian Genome Research Facility in Melbourne. SNP data processing analysis was done by Michelle Gaither and Samuel Greaves at the Genomics and Bioinformatics cluster at the University of Central Florida. The final samples retained for analysis were 72. For the morphology, we modified Foskin's and Lowe's measurements to fit seadragons and added body depth to it. Overall, we did 15 measurements on adult individuals. As you can see, we had various tools that were held above or below, or just very close behind an individual seadragon. Each time we took between 4 to 10 high-resolution photos to get the best shot. We used ImageJ for measurements, and this was done by Katie Mills in Melbourne. Now to the results. So we had 19,608 loci, of which 7,462 were variable and used for analysis. As you can see from our structure analysis, we were provided with 4 distinct populations: Central New South Wales in red, Southern New South Wales from Eden in orange, Tasmanian population in light blue, and the Victorian population in dark blue. The AMOVA also strongly supports these 4 groups of populations, as you can see from the FCT and overall FST. They were all highly significant. These results do support previous mitochondrial DNA results of distinct populations. We can also see there is some evidence of potential interbreeding among individuals from central New South Wales and southern New South Wales and Tasmania. But as you can see, all Victorian individuals were highly distinct to the eastern populations, so no mixing. All these results were further supported by our maximum likelihood tray, our discriminant analysis, and PCA. It is possible that over an evolutionary time frame, the interbreeding has been facilitated along the east coast by the Eastern Australian Current, which could transport seadragons that are rarely washed offshore between these distant populations. A little bit more about that later. Our pairwise FSTs also support our results of 4 distinct populations, as you can see from the high FST values here. For the genetic diversity, individuals from New South Wales and Tasmania were lower compared to the Victorian population. In contrast, inbreeding coefficient was lower in Victoria compared to the FIS of New South Wales and Tasmania. So we also ran isolation by distance and looked at an open connection, so no land bridge, as you can see here, and a closed connection with a land bridge. As you can see, this result also supported strong population structure, especially between East Coast populations of New South Wales and Tasmania, which are the grey circles. Compare that to the triangles, which are the Victorian comparisons. Now, high site fidelity combined with lack of a dispersal phase in juveniles and also their weak swimming ability most likely resulted in the highly genetically structured population of sea dragons. The Bassianismus here acted as a barrier to marine dispersal about 25,000 years ago during the last glacial maximum, and this may have prevented mixing between the East Coast populations from here and the Victorian populations, and thus providing the conditions for the Victorian weedy sea dragons to diverge in isolation. And that's the East Australian current that might have carried individuals from the coast along to Tasmania. To our morphology, over half out of the 15 measurements, so 7 out of the 15, all scaled by total length, this did distinguish New South Wales individuals from Victorian sea dragons. So these are 3 measurements from the head and neck region. That's from here to here, this area here. These are the 3 plots. 2 from the main body, from here to there. They distinguished between New South Wales and Victoria. By the way, Victoria is blue and New South Wales is orange in this case, and black is Tasmania. And lastly, the 2 body depth measurements. Where possible, we did distinguish between females and males. So the open circles or triangles are females and the closed ones are males. We had only a few females, but we did indicate them separately by location in these biplots for body depth, and they showed minimal influence. Okay. When we looked at the linear discriminant analysis for the 3 populations we measured, we had a 96% correct classification for all individuals into 3 groups: New South Wales, again in orange, Tasmania in black, and in blue is the Victorian, and you can see the centroids here as well. While the PCA was not so clear, because it was mainly driven by length, by all the length measurements. All right, so in summary, we have 4 highly distinct populations along the temperate coast of Australia. On the East Coast, we have the 2 distinct populations in New South Wales, as you remember, Central New South Wales and Southern New South Wales, and the Tasmanian population from the East Coast of Tasmania. And to the south, we have the very distinct Victorian population that does not mix with any of the Eastern Coast populations. The total isolation of Victorian population is very important. Since the opening of the Basin Isthmus about 14,000 years ago, there was lots of opportunity for gene flow. So the question now is whether the Victorian sea dragons are reproductively isolated. Please also remember sea dragons from Victoria are primarily used worldwide for the aquarium trade. So we suggest that the Victorian population be recognized as a subspecies and as such provided with a specific conservation management, especially in light of its use in the aquarium trade worldwide. And as you know, as climate change already impacts kelp and seaweed habitats, along the east coast of Australia. This will also affect weedy sea dragons in the future. Thank you, and if you have any questions.
Thank you very much, Selma. There's time for a question or two if people want to unmute, or I'll look at the chat screen.
Hey, Selma. Hello. I've observed that, thank you for a wonderful speech and your nice work. I have observed that the thing that the molecular data and the morphological data are not from the same individuals. Is that right?
No, in some cases there are, and in some cases they are not. You're right there. Because we took DNA samples only for 3 years, and we did more measurement because we didn't have enough measurements. We did much more measurements in 2019 in our last year.
Okay, and I wonder.
Yeah.
I wonder and results about the mitochondrial DNA data of the population Victoria, because you are considering it maybe a subspecies, right? And
that's what we would suggest. Yes, from the genomics.
Yeah,
they are very, very different. Now, I have to say, we only had 20 individuals for the morphology from each of these areas. What should be done is actually do many, many more if possible, and also do more morphological, other morphological measurements to see if this is actually really heavily supported. Plus, the habitats are different. So on the East Coast, most of the sea dragons live in kelp, whereas in the Mornington Peninsula, they are on seaweed and more on seaweed. So it's a little bit different. So that's also an issue we want to look at.
Okay, thank you.
Thanks
Hi, can I
much.
also ask a question?
Yes, of course.
Sorry.
Hi,
Hi, thanks so much for that talk. I really enjoyed it. I was wondering, so you're suggesting that there may be reproductive isolation. Have you, beyond looking at population structure and grouping individuals, also looked at potential signatures of migration or gene flow?
At this stage, that's what we need to do next, and look at behavior, and also especially the reproductive isolation, if that's happening. Because the thing is, we can't do this on the New South Wales sea dragons at the moment. You can't collect them. The only ones collected are from the Flinders area. So everything in aquaria is from that area, and all the behavioral stuff can only be done to them with those, and see how gene flow is. But we do think if you put, for example, if you would put sea dragons from Sydney and from Victoria together, what would happen? We don't know. Are they isolated? Will they pair or not? You know, that's something to be, I don't know if it's possible to get permits for that, but that's something to be looked at. But the data so far shows there is no mixing at all. So the genomics have no mixing at all. Not one single loci was seen in the Victorian population.
All right, thanks very much for your questions, and also Selma for your talk. I think your dog had a question there at one point.
Yes, yes.
Very good. So